Okay, class, this is the Vision Notes Part 2 on Sensation and Perception. Most of the information is taken from the Myers textbook, page 128 to 133. I would fill out page 8 in your packet. What this is going to talk about is visual processing. It's going to talk a little bit about the visual cortex and how we understand color in our vision and vision. Okay, so the last presentation talked about the different structures of the eye. This is just a little recap. Okay, when light enters the eye, light is the stimulus. Okay, it's going to go through the cornea into the pupil. Okay, the iris will expand or constrict, okay, letting light in. The lens then will focus the image onto the retina. Okay, the retina, the eye transduces the characteristics of light into neural signals that the brain can process. This transduction happens in the retina. Okay, so in other words, what this means is transduction is taking place on the retina, or in the retina. Transduction is when it changes from light into a chemical or into a neural impulse. The retina has rods and cones. Okay, a recap from the last presentation. Rods allow us to see black and white, and they're in our peripheral. Cones allow us to see color. Okay. After it reaches the retina, the retina has bipolar cells on them as well. Okay. As a part of the retina, the bipolar cells is the communicator or the link between the rods and the cones, the photoreceptors, and the ganglion cells. So what's going to happen is once it changes into the impulse, okay, through the rods or the cones, the bipolar cells then is going to take that message and send it to the ganglion cells. Okay, the bipolar cells transmit signals from the photoreceptors to the ganglion cells. So in my notes, I would have the process of going from the retina to the photoreceptors, which you know are the rods and the cones, to the bipolar cells, to the ganglion cells. Okay, the ganglion cells then are on part of the retina. The ganglion cells take the information to the optic nerve, okay, and then to the brain. Specifically, it stops at the thalamus first. Okay, so after the ganglion cells take the message to the optic nerve, then to the thalamus helps direct the message to the visual cortex. Okay, so I would make sure from the first slide to the second slide you write down the pattern, okay, or the order in which visual processing takes place. Okay, the occipital lobe, this is from Unit 2. Okay, the occipital lobe is a portion of the cerebral cortex. Okay, it's in the back of the brain. Okay, it helps with visual functions. And the part of the occipital lobe we're going to look at is the visual cortex. The visual cortex, okay, is where sight is registered. It receives written words as visual stimulation. It's at the back of the occipital lobe. So this again is a recap from Unit 2. The visual cortex. In the visual cortex, the brain begins working by transforming neural impulses into visual sensations of color, form, boundary, and movement. So once it gets to the visual cortex, it's actually starting to process those neural impulses. So perception is starting to take place. Different parts of the visual cortex are used to identify different images. Okay, so you can see the top portion of the visual cortex is kind of like a turquoise. That's where maybe houses and chairs are processed. You have the blue, okay, more chairs are being processed. Okay, you have faces in the lower part of the occipital lobe, and then you have the houses all the way in the little green is at the bottom part. Okay, feature detection. David Hubble and Torsten Wiesel in 1979 demonstrated that neurons in the occipital lobe's visual cortex receive information from individual ganglion cells in the retina. So basically what happens is there's specific parts of the visual cortex that are responsible for certain behaviors. Cells that have the ability to respond to the scene-specific feature, so they're called feature detection cells. Okay, they may help you see particular edges, lines, angles, and movement. Okay, they watch different areas in the brain on an fMRI respond to images and then process them in different areas. So if I showed you a picture of a clock, okay, that maybe was moving, okay, some of the cells might be looking at the lines, some of them might be looking at the angle, some of them might be watching the movement of the clock, okay, and the different parts are responsible from the future detection cells. Damage to certain areas would be damage to processing different components. So you may be able to see the lines on the page, but you might not be able to understand that the clock is moving. All right. Parallel processing is another visual component that happens in the visual cortex. Our brains are able to process many things in a scene at once. This is called parallel processing. Okay. The simultaneously process of several aspects of a problem, they do it simultaneously. The brain begins to divide up the scene into subdimensions such as color, movement, form, and depth. Okay, so it's processing a lot of things at once, such as color, motion, form, depth, all in those different areas. Okay, it's happening at one time. All right. We construct our perceptions by integrating the separate but parallel work of these different visual teams. Okay, so parallel processing and facial recognition. To recognize a face, for example, the brain integrates information that the retina projects to several visual cortex areas. Okay, we just saw that in the feature detection cells. All right, and it compares it to stored information that enables you to recognize the image, and then it's using parallel processing to process a lot of things at once. Okay. It takes about 30% of the cortex for facial recognition. So it takes a lot of the brain to understand the different visual processing that's taking place. 10 times the brain area devoted to maybe hearing something or recognizing somebody's voice. Okay. Miss H case was the case study that was in your book. She had a stroke which damaged the back area of her brain, some parts of the visual cortex, specifically the area that controlled movement. Okay, so the feature detection area, cells that controlled movement, she lost that ability because of the stroke. So when they were pouring tea, okay, which you understand that pee would look as like a fluid and it would be moving, she didn't understand that the tea was pouring into the cup. So to her, it looked like the tea was constantly frozen. Okay, so because she didn't have that ability to understand movement, items would look as if they were frozen or freeze. Okay, color is really a mental construction of how we process the different light spectrum. We can discriminate some 7 million different color variations. Okay, 1 in 50 people are genetically deficient to certain colors, and we'll talk about color blindness in a little bit. Okay, there are different theories on color vision. All right, the first one we're going to look at, okay, is the Young-Helmholtz or trichromatic theory. Some situations you may see it titled as Young-Helmholtz. Others you may see it as trichromatic theory. You need to know it's the same theory of why we see certain color. So people came up with ideas of how we interpret or understand color. So in the 19th century, Hermann von Helmholtz, built on the work of Thomas Young, okay, came up with this theory. Okay, he believed that the retina contains 3 different color receptors: red, green, and blue. Okay, combinations of the stimulus of the various light produce then the color spectrum. Okay, so he believes that if we kind of combined red and green, it's going to make like a yellow component, and then if we combined the light spectrum of red and blue, it's going to make a purple component. If we combine all of them, since we're looking at light, it's going to create a white color. Okay. Color blindness then is the lack functioning of red. You might lack that color of red. You might lack blue, okay, or you might lack the ability to understand green. Okay, so then you struggle with making the combination of that color. Okay, so if somebody were color deficient in blue, all right, they would be able to understand red and green and yellow, right? But they may have deficiencies in understanding like the indigo, the blue colors. Okay, so we are trichromatic if you understand or have all three of those. Okay, if you're only having one, you're going to be monochromatic, or if you only have two colors, you're going to be dichromatic. Okay, color blindness, not everyone sees color in the same way because some people are born with a color deficiency. While some people can see no color at all and are totally colorblind, that's really rare, so they don't have any red, we just did it, red, green, or blue, okay? But most people are just missing one of them. More common is color weakness, where people have a hard time distinguishing between the certain colors. Okay, so this is where I noticed a student may be potentially colorblind. Okay, if hopefully you can detect that there's a number within this, these a bunch of the different circles. Okay, or you can notice that there's another number in the different circles. If you were colorblind, you might not be able to recognize the different numbers. Okay, that there are numbers in there. Okay. The opponent process theory is another theory of color and how we see color. Herring proposed that we process 4 primary colors combined in pairs of red, green, blue, yellow, and black and white. Okay, so we have our 3 basic strong blue, green, and red, which we talked about before, the cone. Okay, and then the ways that we process the primary colors is when we combine the certain colors. So blue and yellow, red and green, and black and white. And you need to know, okay, what are the paired colors? Okay. So what happens sometimes is after you're soaking up your eyes in like a green, you're soaking up your eyes with the green exposure. Okay, sometimes then if you look away on a blank screen, you present an after image, which your cones really want to use that red image that it has. So you're going to start seeing the color red. Okay, same thing, blue opposes yellow or black opposes white, and this is known as the after image effect. Okay, so if you want to look at the American flag, okay, you're looking at the different images here. I usually stare at the corner star, and I stare at it for 30 seconds. Okay, so you can freeze this presentation for 30 seconds. All right, and then when you want to press play, you're going to go to the next screen, and supposedly right after this, you're going to see red, white, and blue flag because that's the after image effect. Okay, now when you're projecting your eyesight on the next blank slide, okay, you should see the American flag. If you see it in red, blue, and white, then the after image has impacted you. Okay, so in recap, we looked at visual processing. You should be really good at understanding that light comes in through the lens, through the pupil, okay, and then projects eventually onto the retina. If the retina goes from the cones to the rods, and you know the difference between bipolar and ganglion cells. Okay, you should understand the difference in the visual cortex. Some of the things that happen is parallel processing, and you have feature detection cells. And then you should know the last 2 theories on color, the opponent process theory and the Young-Helmholtz or trichromatic theory.